Hello, my name is Matthias Maiwald and I'm a senior consultant in microbiology at KK Women's and Children's Hospital in Singapore and also an adjunct associate professor in microbiology at the National University of Singapore. The title of this talk is When Evidence is Not What It Seems, Complexities in the Assessment of Skin Antiseptics. In this presentation, I will talk about the complexities involved in the assessment of skin antiseptics. Next, I have no conflicts of interest to disclose, and the views expressed are solely my own. The format of this presentation will be like a question and answer session. We have 3 topics, and each topic has several questions that I will address. Topic 1 is the forgotten role of alcohol in antisepsis. The first question is, why did you decide to do your meta-analysis that was published in 2012 in PLOS ONE? Now, around 2010, I started to notice inconsistencies in the literature on skin antisepsis. What happened was that the clinical studies and then meta-analyses compared clinical outcomes after skin antisepsis with chlorhexidine alcohol combinations versus aqueous povidone iodine, and concluded that chlorhexidine is better than povidone iodine without mentioning the alcohol component. This did not make sense because chlorhexidine alcohol is a dual active preparation with 2 effective antiseptics, whereas aqueous povidone iodine is only 1 effective antiseptic. So this was an unequal comparison. It is well known from many decades of antiseptic research that alcohols are among the most potent skin antiseptics but have no persistent activity on skin. On the other hand, chlorhexidine or povidone iodine are less powerful, but they have persistency on skin, which is greater for chlorhexidine than for povidone iodine. Now, other colleagues in the field also noticed the problems in the literature. And initially, we wrote a few letters to the editor in response to some of these articles. But it became very obvious that the authors of these articles did not understand that alcohol in itself is a very effective antiseptic. It seemed that authors viewed alcohol as a carrier substance or solvent for chlorhexidine and viewed chlorhexidine as the only active component in the mix. So we decided to investigate this phenomenon more closely. The next question is, what were your study methods and results? So we decided to do a systematic literature review and meta-analysis, and we asked the following questions. What is the factual evidence for chlorhexidine alone or for chlorhexidine in combination with alcohol in skin antiseptics? How common is it that authors say that outcomes are from chlorhexidine alone when in fact a combination of antiseptics has been used? Then, could this phenomenon have unjustly influenced evidence-based guidelines in favor of chlorhexidine? We used the following systematic review methodology. We did an exhaustive literature search for clinical trials and systematic reviews, looking at chlorhexidine-containing antiseptics versus other antiseptics. We looked at 3 applications. First, skin antisepsis before blood culture collection. Second, skin antisepsis before vascular catheter insertion. And third, surgical skin preparation. We also looked at the attribution of study outcomes, meaning whether authors attributed study outcomes from chlorhexidine alcohol to chlorhexidine alone. We performed meta-analysis, meaning mathematical calculations of the factual evidence for chlorhexidine versus other antiseptics, mainly that was povidone iodine. And we did a review of tertiary literature, meaning guidelines and recommendations, to see whether they contained unjustified recommendations for chlorhexidine. To illustrate our criterion for attribution, imagine a typical scheme of a clinical trial where you have trial arm A, and this trial arm A used 2 different antiseptics in combination, and this creates trial outcome A. And on the other hand, there is trial arm B, which used only a single antiseptic and created trial outcome B. So We looked at whether authors made conclusions such as trial outcome A is solely caused by chlorhexidine, when in fact chlorhexidine and alcohol had been used in combination, or at conclusions to say that chlorhexidine is better than povidone iodine, when in fact this is impossible to tell because chlorhexidine had been used together with alcohol. The overarching conclusion was often that the evidence supports chlorhexidine, when in fact this was impossible to be concluded. This means the study authors made conclusions that could not be made. What were our main findings? First, we found good evidence that chlorhexidine and alcohol is better than aqueous povidone iodine for blood cultures, for catheters, and for surgery. However, the evidence for chlorhexidine plus alcohol versus iodine when combined with alcohol was either equal or inconclusive. We found no evidence supporting the use of chlorhexidine alone for blood cultures and surgery. We found some evidence that aqueous chlorhexidine is better for catheters than aqueous povidone iodine, but overall the evidence for chlorhexidine and alcohol combinations for catheters was better. Incorrect attribution of clinical trial outcomes was very common. An average of 34% of articles had incorrect attribution, and when ambiguous or unclear attribution was combined with incorrect one, then an average of 54% of all clinical trials and systematic reviews were not clear about what caused trial outcomes. Furthermore, information on antiseptic composition was often incomplete or missing. Only 35% of all clinical trials and systematic reviews both correctly listed the antiseptics that had been tested and correctly attributed efficacy to the components used. What this means is also that about 65% of clinical trial and systematic review authors were not fully aware of what they had actually studied. We published our findings in the journal PLOS ONE in 2012, and what you can see here is the title page of the article. So the next question is, do these misinterpretations favor a specific antiseptic agent? And I would say yes, these misinterpretations favor chlorhexidine. This is because the factual evidence for chlorhexidine alcohol was incorrectly perceived and interpreted by many people as evidence for chlorhexidine. But in most cases, the evidence was obtained under conditions where it was impossible to separate the effects from alcohol from the effects of chlorhexidine. The next question was, did these misinterpretations affect guidelines and clinical practice recommendations? And the answer is yes. The perception that chlorhexidine is the only active agent when in fact the combination was used created an impression that chlorhexidine is the in-antiseptic that is favored by evidence. In our article, we also looked at tertiary literature, meaning at evidence-based guidelines and clinical practice recommendations. We found several examples of unjustified recommendations for chlorhexidine when in fact the evidence was either not clear or when the evidence supported chlorhexidine alcohol combinations. The next question is, what happened in the literature after your 2012 PLOS ONE article was published? Now, this is a mixed picture. Some articles referred to our article, correctly assessed the skin antiseptics and made appropriate recommendations for chlorhexidine alcohol combinations when this was correct, or made recommendations for either chlorhexidine alcohol or iodine alcohol when this was indicated. So it seems that the understanding of alcohol as an important antiseptic is increasing. One example of correct recommendations is the 2014 Society of Healthcare Epidemiology of America and Infectious Diseases Society of America paper on preventing surgical site infections. The authors correctly recognized that alcohol is an important component for surgical skin preparation and also recognized that there was insufficient evidence to recommend chlorhexidine alcohol over iodine alcohol. On the other hand, there is still in the literature a considerable number of articles making the same previous mistake. Unfortunately, there are still unjustified recommendations for chlorhexidine containing antiseptics in situations where this is not clear. Topic number 2 is pitfalls in evidence of skin antiseptics. And the first question is, what are common pitfalls and misinterpretations in the assessment of chlorhexidine and alcohol? Now, we summarized some of these pitfalls and misinterpretations in 2 articles. One commentary style article in the Journal of Antimicrobial Chemotherapy in 2014, and another letter to the editor in Infection Control and Hospital Epidemiology in 2013, and in both we summarized some of these pitfalls and issues. There are 3 main sources of errors and misinterpretation in the literature. The first type of error occurs when microbiological efficacy testing is done without adequately validated neutralizers or completely without neutralizers. Neutralizers are to prevent continued action of the antiseptic after the end of testing. Now, all antiseptics require appropriate neutralizers, but chlorhexidine is more affected than other antiseptics because of its mode of action. Unfortunately, there are a number of articles in which neutralization has not been done appropriately, and this includes clinical trials with microbial skin counts. Second, the pitfalls also include the incorrect attribution of efficacy from combination antiseptics to a single ingredient that we discussed earlier. This creates a false perception that only one antiseptic is effective, when in fact a dual active combination has been used and most likely caused the trial outcomes. Third, when looking at the published literature, one can find a number of statistical inaccuracies and errors. There are numerous ways in which this can happen. We have seen quite ordinary miscalculations of statistics. For example, trials where the results were insignificant, but the authors published that they were significant. Then we have seen inappropriate groupings of trial arms for statistical calculation. For example, in one prominent trial, the results were not significant, but when the trial authors grouped together the antiseptics in such a way that chlorhexidine was significantly better than the other antiseptics. Then some trials engage in selective reporting. Selective reporting is when outcomes are reported that are not consistent with outcomes that make clinical sense, or when outcomes are different from what the authors said they would do during the trial planning. Note that trial plans should be made in advance and are usually published on trial registry websites. So in my opinion, it always pays to look at how well the authors have done their statistics and their trial reporting before believing in the published literature. Unfortunately, these errors and misinterpretations affect chlorhexidine disproportionately more commonly than other antiseptics, and this is often in favor of chlorhexidine. The overall effect is that there is now a perception of chlorhexidine as the in-antiseptic, while chlorhexidine is most certainly a very effective antiseptic. This is actually disproportionate to its real benefit. Slide 13 is just to explain briefly the principle of neutralization. Some antiseptics continue to act even after the end of antiseptic exposure. To prevent this, it is necessary to add neutralizing substances to the sampling medium so that the action on microorganisms is stopped at the point of sampling. This is a general requirement in all standardized microbiological tests, such as the US tests and the European EN tests. Failure to neutralize appropriately can result in false positive efficacy assessments. As mentioned, this effect is particularly pronounced for chlorhexidine. So the next question is, how do you see the role of microbiological antiseptic testing? Now, microbiological antiseptic testing has a history of nearly 100 years, and this has helped the infection control community to assemble a very large and very valuable amount of information. Unfortunately, this information is not available in a single or just a few articles, and instead it requires reading from many different sources to obtain a good picture. So what are the principles of microbiological testing? There are a few different types of tests. First, there are tests in reagent tube format in vivo. A classical one is the suspension test, in which the organisms are exposed to the antiseptic, and after certain times it is tested how many organisms survive. Second, after the in vitro tests have shown efficacy, there are tests under simulated practice conditions, such as tests on real hands and on real skin. These tests provide a realistic scenario on how the antiseptics are applied, and the tests are done on healthy volunteers. The experimental conditions have been standardized, and there are both US standards and European standards available. There are two major sets of testing standards, the US standards and the European EN standards. Not all applications are represented in standards. For example, there is a US ASTM test for skin, but there is no EN test for skin. But this can be supplemented by national tests. For example, the German VAH, which is Association for General Hygiene, has devised a test, and the VAH test works quite well on skin. So what are the advantages and disadvantages of microbiological testing and of clinical trials? So first, microbiological testing provides indirect information. It does not test real clinical outcomes, and there is a risk that the real clinical outcomes may differ. However, the history of antiseptics and clinical experience shows that the results correlate quite well with the clinical performance. Detailed testing is possible, and it is common that manufacturers do extensive testing during product development. Microbiological tests are also a common requirement for product registration, and you should be able to ask a manufacturer what is the performance of your product in standardized tests, and they should give you this information. And another advantage is that in microbiological testing, there is no risk for patients from real infections if a product is not performing well. On the other hand, clinical trials measure real clinical outcomes. They can be used for systematic reviews and meta-analysis, and therefore provide the best possible evidence. However, each single test requires hundreds or thousands of real patients, and this poses logistical challenges. There is also a risk of real infections for patients, and if we think of catheter-related bloodstream infections or surgical site infections, they can be clinically severe. So the question is, are there ethical implications? And I would say yes. I have seen several clinical trials where it was clear before the trial that one of the competitors was likely superior to the other based on known microbiological performance. None of these trials released the microbiological test results. Some trials had potentially serious infections as clinical outcomes. As expected, more patients who were treated with the inferior antiseptics had adverse clinical outcomes. And the question arises, is this ethical? Personally, I must say I don't have an answer to this question, but I think that these questions should be discussed in the wider community. There is also a principle called clinical equipoise, whereby there should be genuine uncertainty at the beginning of a trial about whether a treatment will be beneficial before entering a trial. If we are doing a trial with one microbiologically very strong antiseptic and one microbiologically weak antiseptic, I don't think that we have clinical equipoise. What I should add is that I have not seen any clinical trial results that are contrary to the known microbiological properties of antiseptics, and this provides further support for the usefulness of the information coming from microbiological testing. So the next question is, what other aspects need to be taken into consideration? And I would say there are indeed a few other aspects that are relevant. First, one should consider the physiological and functional requirements of skin antisepsis. This becomes apparent if you look at the scheme in the slide. Blood culture collection requires strong and immediate microbicidal action before puncturing the vein, but after the blood is taken, there is no need for persistency at the skin sites. Alcohols are very suitable for this purpose because they have strong immediate activity. On the other hand, for surgical skin antisepsis, one needs strong and immediate activity before incision, and one needs low numbers of organisms at the surgical site for a few hours of an operation. So surgical skin antisepsis will benefit from alcohols, but one wants to add some persistency. Then vascular catheters are remarkable in that they remain in the skin site for many days, not just for minutes or hours. That means vascular catheters have special requirements. Logically, they will benefit from strong and immediate activity before insertion, and that is what the alcohol does. But then there is an advantage from persistent activity on skin. Chlorhexidine is well known for its persistency on skin, and that is where it has shown a clinical advantage. So, chlorhexidine alcohol combinations are currently the best choice for vascular catheter sites and are supported by strong clinical evidence. But this does not necessarily mean that it performs equally well in other clinical applications that have a different physiology. There is an interesting aspect concerning povidone iodine. Povidone iodine is an organic iodine complex that releases free iodine, and the free iodine is what kills microorganisms. The availability of free iodine is not just dependent on the total iodine content, but also on how well stabilized the overall solution is and what pH and other components it has. There can be quite different microbiological performance between different povidone iodine preparations. Now, the implication is when you have a clinical application or when you do a clinical trial, would you expect the same clinical outcomes from preparations that perform microbiologically quite differently? I suspect not. So the outcomes of clinical trials may depend on what povidone iodine preparation has been used. One would want to make sure before a clinical trial that one is using a microbiologically well-performing preparation. Otherwise, the clinical trial results may not be representative. So the next question is, what do you think is necessary for antiseptic assessment? Now, I can see some confusion in the literature and in the various recommendations on skin antisepsis, and this is often a result of an incomplete picture whereby important aspects have not been taken into account. One important thing to consider is that evidence for skin antiseptics does not solely arise from clinical trials out of a prior vacuum of information. This sounds trivial, but it is very important because otherwise we would be overlooking very valuable information sources. If you look at a typical drug development process, such as the US FDA drug development process, then you realize that there are many other highly valuable information sources that come before a clinical trial, such as laboratory testing, animal experiments, etc. For example, the European Committee on Standardization, Technical Committee 216, has devised a logical stepwise approach to antiseptics assessment that proceeds from pure in vitro testing towards clinical applications. The US FDA thinks along similar lines but has not formally spelled out a stepwise process. So, I am proposing that we must have a more holistic approach for assessing antiseptics. So we need to take into account the knowledge of basic antiseptic components. What are we using and what is known about these ingredients? We need to look at the clinical and functional characteristics of antiseptic applications. What are the characteristics of the intended applications? Do we need persistency? is there any use on mucous membranes? Then we need to have results from state-of-the-art microbiological testing according to recognized standards, for example, the European EN or US ASTM standards. This is a common requirement for product registration. And in my opinion, no product should enter clinical trials without detailed microbiological test results available. And then we can proceed to clinical trials. These should be done according to state-of-the-art clinical trial standards, and we need to be cognizant of the ethical implications. For example, could there be adverse outcomes for patients if we use microbiologically inferior antiseptics? Finally, we need to consider biological plausibility. Do the findings fit with what we know, and do the findings make sense? So, as I said, we need a holistic assessment of antiseptics. The next topic is antiseptic recommendations in the new WHO surgical guidelines. And the first question within that topic is, are the guidelines relevant for developed countries, especially for France? Now, as far as I am aware, the guidelines are meant by WHO to be global guidelines, and so they are meant to be relevant for both developing and developed countries. But how relevant they are depends on a country's framework in place for antiseptic testing and antiseptic assessment, and whether this is better than how the WHO did their assessment. I cannot comment much on France's antiseptic assessment and approval framework because I'm not familiar with it, but ideally a country should have stringent antiseptic testing standards and requirements in place. The next question posed was, are the guidelines really evidence-based? Now, the guidelines are certainly evidence-based in the sense that the WHO has applied a formal process of evidence assessment before formulating its recommendations. The question then is, how well has the evidence been assessed, and how good is the quality of this evidence assessment? Now, if you look at the WHO recommendation for surgical skin preparation, as in this slide, the recommendation is for an alcohol-based antiseptic solution based on chlorhexidine. Please note that this is listed as a strong recommendation with only a low to moderate quality of evidence. Note also that it was fairly clear from other sources and recommendations that the skin antiseptic should be alcohol-based. So the remaining question was now whether it should be a chlorhexidine alcohol or iodine alcohol combination. So the WHO did a meta-analysis of chlorhexidine alcohol versus iodine alcohol studies, which you can see here. And if you look at the bottom, the diamond shape at the bottom is left of the middle line and shows that the results are significant in favor of chlorhexidine alcohol. This slide shows the meta-analysis on which the WHO recommendation is based in more detail. Look at the characteristics of the included studies. 2 of the studies had no specified alcohol content in the chlorhexidine alcohol or the iodine alcohol preparations. 1 study had an isopropanol content of only 23% in the iodine alcohol trial arm, and with such low concentrations or with unknown concentrations, one simply cannot assume a participation in the antiseptic process, meaning that it is impossible to say which antiseptic ingredients contributed to microbicidal activity. Also, if you look at the second trial from the bottom, the WHO exceptionally included 1 study from 2016, which is the Tooley et al. 2016 study, and this was published after its initial literature search for studies. This slide shows a reanalysis of the meta-analysis of chlorhexidine alcohol versus iodine alcohol. What I have done is I have excluded all trials with unknown or insufficient antiseptic content and have only included trials with known content where the contents were within known antimicrobially active concentration ranges. I have also found additional trials that were not included by WHO. Notable is one large trial by Nguy published in 2015. This trial was published in 2015 before the trial Tooley et al. 2016 that was included by WHO. So the WHO should have been able to find it. And when the meta-analysis is recalculated, the results are not statistically significant anymore between chlorhexidine alcohol and iodine alcohol. The next question is, are these guidelines premature? And I would say yes. In my opinion, the WHO's recommendation for chlorhexidine alcohol for surgical skin preparation is premature. As I have shown in my updated meta-analysis, when one includes only trial with appropriate antiseptic ingredients, which is what should be done, the results are no longer statistically significant. Other major guidelines for surgery, including the new 2017 CDC guideline, continue to recommend alcohol-based antiseptics with either chlorhexidine or iodine. Now, the evidence in the future may eventually turn out to favor one over the other, and we may see either chlorhexidine alcohol or iodine alcohol favored in the future, but this is not the case at this point in time. The next question is the efficacy of chlorhexidine and povidone iodine similar or comparable? Now, there is no clear answer to this question. There are differences between chlorhexidine and povidone iodine. Chlorhexidine overall has greater persistency on skin, and that is probably a good explanation why chlorhexidine alcohol performs very well for vascular catheters. On the other hand, povidone iodine is an effective antiseptic, and its microbiological efficacy depends on the formulation, and not all preparations are the same. An important question is the comparison of chlorhexidine alcohol versus povidone iodine alcohol for other applications than catheters, and this is where things are still undecided, particularly in surgery. So this slide shows an interesting microbiological comparison of chlorhexidine alcohol versus povidone iodine alcohol in a skin test. If you look at the values underneath the baseline in the picture, you see the results, and the red is iodine alcohol and the green is chlorhexidine alcohol. So it seems that povidone iodine alcohol have initially greater microbial reduction than chlorhexidine alcohol, and this suggests the possibility of additive or synergistic activity. However, due to the greater persistency of chlorhexidine, the results further down the track at the 6-hour time point become equivalent. It is also clear that more data are required. We should not rely on a single or just a few studies, and it would be a big benefit if more comparative data from standardized tests are published and become available for the infection control community. These data should be a prerequisite for clinical trials, and I suspect that the clinical and microbiological data would nicely supplement each other. Coming to a conclusion, and in closing, it is clear that more work must be done and more effort must be undertaken to achieve appropriate and factually correct assessment of antiseptics. As I said before, we need a more holistic approach that includes knowledge of the basic of antiseptics, physiology of applications, standardized microbiological testing, clinical trials, and the principle of biological plausibility. And at this point, I would like to come to the conclusion and thank everyone for their attention. Thank you.